electron transport system and oxidative phosphorylation. Electron transport system and oxidative phosphorylation occurs on the cristae of mitochondria. Inner membrane of mitochondria called cristae. It shows folder-like structures. On inner membrane, mainly 5 complexes are present. They are complex 1, complex 2, complex 3, complex 4, complex 5. Complex 1 is look like a pillar-like structure. It containing flavin mononucleate protein. Complex 1 is also called NADH dehydrogenase. Complex 2 is in spheroidal structure. It present near to the matrix of mitochondria. It containing flavin amide adenine dinucleotide. Complex 2 is also called succinate dehydrogenase. Complex 3 look like a L-shaped structure. Complex 3 is also called cytochrome B-C complex. It contains cytochrome B and cytochrome C1. Complex 4 look like a pillar or heart-shaped structure. It containing cytochrome C and cytochrome A3. It is also called cytochrome oxidase complex. Complex 5 is also called ATP synthase. ATP synthase contains collar and spheroidal head. Collar part present in the inner membrane of mitochondria. It is also called F0 portion. And spheroidal head towards mitochondrial matrix is also called F1 particle. F1 on F0 particles commonly called ATP synthase. These are the 5 main complexes helps in electron transport chain and synthesis of ATPs. During the respiration, cytosol NADHs are formed in the cytoplasm of the cell during the glycolysis. During the glycolysis, 2 cytosol NADHs are formed. These cytosol NADHs enter into the matrix of mitochondria by the diffusion process. During the link reaction Krebs cycle, NADHs also formed. These NADHs are called mitochondrial NADHs. During the Krebs cycle or citric acid cycle, 2 molecules of FADHs are formed. And mitochondrial NADHs total 8 in link reaction and Krebs cycle. These 3 components are involved in the electron transport chain. First, we discussed about mitochondrial NADH electron transport system. Mitochondrial NADH split into 2 electrons, protons and NAD+. Protons and NAD+ are present in the mitochondrial matrix and 2 electrons received by complex 1. When the complex 1 received 2 electrons, at the same time, the complex 1 received 4 protons from matrix. The received 4 protons are transported into IMS, inner membrane space of mitochondria, and the 2 electrons pass to ubiquinone pool. It present between the complex 1 and complex 3. From complex 1, the 2 electrons are pumped to ubiquinone. From ubiquinone to complex 3, not complex 2. Complex 3 received 2 electrons from ubiquinone pool. When complex 3 also received 4 protons from the matrix, and received 4 protons are pumped into IMS of mitochondria. And the 2 electrons pass to complex 4 through the cytochrome C mediator. When the cytochrome C pumped the 2 electrons to complex 4, the complex 4 received 2 electrons. At the same time, 4 protons also received from matrix by complex 4. From these received 4 protons, only 2 protons are pumped into IMS. and remaining 2 protons, electrons combines with of oxygen molecule convert into or formation into one water molecule in the matrix of mitochondria. During the mitochondrial NADH electron transport chain, total 10 protons are pumped into IMS. From complex 1, 4 protons, complex 3, 4 protons, complex 4, 2 protons. Total 10 protons are pumped into IMS with the mitochondrial NADH electron transport system. And cytosol NADH is also present in the mitochondrial matrix. The cytosol NADH split into electron, protons, and NAD+. Electron received by complex 1. When the complex 1 received 1 electron from NADH splitting, the 2 protons are pumped into IMS and 1 electron pumped to ubiquinine. It present between the complex 1 and complex 3. Ubiquinine received electron pumped to complex 3. When the complex 3 received electron from ubiquinine pool, it received 2 protons from matrix and that received 2 protons pump into IMS. Then the electron passed to complex 4 through the cytochrome C mediator. When complex 4 received electron from complex 3, the complex 4 also received 2 protons from matrix and received 2 protons are pumped into IMS. Electron pump into matrix of mitochondria. Through the cytosol NADH electron transport chain, from complex 1, 2 protons, from complex 3, 2 protons, from complex 4, 2 protons, total 6 protons are pumped into IMS. And another one is FADH2 electron transport system. FADH also split into 2 electrons, protons, and FAD+. The protons and FAD present in the matrix of mitochondria and 2 electrons received by complex 2. When the received 2 electrons by complex 2, the 2 electrons are pumped into ubiquinine pool. The ubiquinine pool pumped 2 electrons to complex 3. When the complex 3 received 2 electrons from ubiquinine or ubiquinine pool, it received 4 protons from matrix. The matrix 4 protons are pumped into IMS. From complex 3, 4 protons are pumped into IMS and the remaining 2 electrons pump into complex 4 through the cytochrome C. When complex 4 received 2 electrons, it is also received 4 protons from matrix of mitochondria. And the 2 electrons received 4 protons. From that 4 protons, 2 protons, only 2 protons are pumped into IMS. Remaining 2 protons and the 2 electrons combines with of oxygen molecule to make one water molecule in mitochondrial matrix. It is the 3 types of electron transport chain in the mitochondria crystal. After completion of mitochondrial NADH electron transport chain, cytosol NADH electron transport chain, FADH electron transport chain. Then, after completion of these 3 electron transport systems, matrix protons are pumped into IMS. In IMS, inner membrane space, proton concentration is very high. According to diffusion, high concentration of ions moves to low concentration. Mean inner membrane protons are moved to matrix of mitochondria, but the inner membrane cannot helps in the movement of the proton transportation. But in some areas, ATP synthase particles are present. Through these ATP synthase, 3 protons are moved from inner membrane space to matrix. The 3 protons are generate some amount of energy. That energy is helps in binding of ADP molecule with inorganic phosphate to make ATP. So ATPs are synthesized in the matrix in presence of oxygen. So this process is also called oxidative phosphorylation. Oxidized phosphorylation, synthesis of ATPs in presence of oxidation reactions or in presence of oxygen. Finally, the movement of protons helps in synthesis of ATPs. The ATPs are synthesized in presence of oxidative reactions called oxidative phosphorylation. In glycolysis, total ATPs, cytosol NADHs are 2. Each NADH produce 2 ATPs. Total from 2 NADHs, 4 ATPs are formed. And direct ATPs are 4 developed. Total ATPs are in glycolysis 8 and utilized ATPs are in glycolysis 2. Net gain of ATPs in glycolysis 6 ATPs. In link reaction and Krebs cycle, mitochondrial NADHs are formed 8. Each NADHs of mitochondrial develop into 8 ATPs. Total 24 ATPs are developed from mitochondrial NADH. FADH are 2 developed during the citric acid cycle or in the Krebs cycle. From each FADH, 2 ATPs are formed. Then total 4 ATPs are formed from 2 FADH. And direct ATPs are 2. Net gain of ATPs in link reaction and Krebs cycle total 30 ATPs. Total net gain of ATPs are 36 ATPs. 1 ATP produced 7.300 calories of energy from glucose. One glucose molecule produced 262.8 kilocalories of energy. It is a oxidative phosphorylation and link reaction in the respiration.